Good evening, everyone. Welcome to day 3 of this international webinar on quality by design. Without wasting much time, let me ask Sai Kishore to introduce the profile of today's person.
Good evening to one and all, and welcome to third day's session and last session of our 3-day international webinar on quality by design, organized by Bapatla College of Pharmacy in association with Education and Pharmaceutical Association. I would like to introduce today's speaker, Dr. Venkat Krishna, who is going to give us a talk on QbD and biology development. Let me give his biodata. Dr. Venkat Krishna obtained his bachelor's degree from IIT Bangalore and PhD from Rutgers University. After working in branded multinational companies such as Research Genetics, Bayer's, Novartis, and Merck, he became an entrepreneur with his own startup, QbD Pharma, which provides scalar, transfer, and validation expertise. He implemented quality by design and process validation using statistical process generic products. He has several US patents related to drug development in small and large molecules. He is an avid reader of Sanskrit literature and supporter of Indian art. Back in 2000, his research on large-scale fermentation and purification of protein under microgravity environment helped NASA during the construction of International Space Station. It is a great privilege to have you today with us, sir. Over to Dr. Venkat Krishna, sir. Sir, now the podium is yours. Please go ahead, sir.
Hello, good
Yes,
morning.
Yeah, we can hear you, sir.
Thank you. I would like to take this opportunity to welcome everyone for the third and penultimate webinar on quality by design in biologics development and manufacturing. Today, we are going to see the most important aspect of what biologics are, especially given the current scenario that we are in, and how researchers and educationalists will be able to provide a development and a successful manufacturing of products that can help in alleviating this global pandemic that we are currently in. So let me start with a little bit of the basics. And I want to let you know that we are here in discussion about large molecules. The previous 2 days in the seminar was strictly related to the small molecule development. Small molecules are very chemically synthesized and have a typical molecular mass less than 1000. Whereas large molecules are proteins. They are biologics. And they have more than 1000 amino acid chain, and they follow a very complex synthesis method. They differ in shape, size, morphology, route of application, and the method of action. So we are talking completely two different entities, small molecule versus large molecule. Now, when you come to proteins, what are proteins? Proteins are in our body that are present that helps with the building blocks. And proteins are made up of amino acids. And there are totally 20 different kinds of amino acids, and whose combination gives complex proteins. And the genetic code for protein synthesis is in the DNA. And some of the major functions of proteins, which you might or you might not know about, are structural proteins such as collagen, contractile proteins such as your muscles for moving action. There are transport proteins that carry oxygen, hemoglobin. There are enzymes which are biologic catalysts, hormones such as insulin, thyroid that activate certain cells and certain organs of your body for critical body functions. And the most important are antibodies. For example, human insulin are produced for treatment of diabetes are large molecules, and these are synthesized and being marketed. Now we come to the concept of biotechnology, which is use of enzymes and microbes to produce products and processes that are useful for human consumption or biological use. And the most important aspect is related to a combination of biology, molecular biology, pharmacology, therapeutic effect and action of drugs, and the underlying chemical engineering principles such as heat transfer, mass transfer, momentum transfer that are associated with concept to commercialization of these biologics. And we need to understand what is the central dogma. The central dogma of molecular biology is DNA is transcripted into the RNA, and RNA is translated into proteins. And proteins are, as I mentioned, very complex, and they do many different functions. So the key to any protein synthesis is stored in the DNA of the organism. Now we come to a novel concept that came up in the late 70s, early 80s called recombinant DNA, where they were able to take a vector, it is called typically a plasmid, a chain of sequences, and they were able to splice it and insert a foreign DNA. And this foreign DNA is polymerized with the vector and introduced into cells, bacterial cells, mammalian cells, to produce the genetic code that is locked in inside the DNA to produce the protein of interest. You're very much familiar with the movie Jurassic Park, where they were able to sequence the DNA stored millions of years ago as a vector and create dinosaurs out of that. So it is possible, and we do have the drug approval that we received. In 1982, FDA granted the approval of human insulin to Eli Lilly as the first therapeutic protein using the recombinant DNA technology. And another product that was approved in 1983 was by Genentech, which is called Activase. As you are all familiar with, Genentech was a small startup based in the West Coast of America, which sold the rights to the recombinant DNA of insulin to Eli Lilly. So for entrepreneurs who are looking for innovative solutions, there are big companies that are always looking to bring in innovative technology to the market. So what are some of the recombinant crops that are being used using the DNA technology? We have the Bt cotton, which is resistance to the bollworms. There is the superphos B that is used for producing opium and alkaloids. And then Monsanto created the genetically modified corn or maize. So these are available, these are being used, and India is one of the largest users of BT cotton because India is number one in textile manufacturing and production. So there is a lot of interrelationship between biotechnology and the commercial and macroeconomic aspect of a country. Now, I mentioned that there were 6 different functions of a protein, and the last one are antibodies. And if anyone is out there and you need to know what your blood groups are, it's typically A positive, B positive, AB positive, and O negative. What does that refer to? That is the antigen and the antibody that is being present in your body to respond to that. So some of them are universal, such as O. Some of them are specific only to A, B, or AB. And certain cases like AB, they do not have either of those antibodies, and they have a higher propensity for diseases. So when you come to the concept of antibodies and MAB, a lot of the new drugs that you see that are getting approval ends with MAB. So what is MAB? MABs are monoclonal antibodies. If you're able to take an antibody from your body and able to produce millions and billions of it that are just the exact replicate clones of what that one antibody is, you're able to call that as monoclonal antibodies. And there are 2 different pathways by which you could synthesize them. And basically, you give an antigen, and there is a response of the body that produces these antibodies. You are able to take those antibodies, fuse it into hybridoma cells, and you amplify these cells. And the output of it is the monoclonal antibody. Another way to look at it is as well, you are able to genetically decode the antibody gene sequence, and you're able to introduce that through recombinant DNA technology as a vector into different kind of cells. It can be bacterial cells, it can be mammalian cells, and you produce the same antibodies in millions and billions of clones and replicates. Now, in this COVID situation, you are very much familiar with a new therapy that is emerging that is called plasma therapy. So you have a COVID patient who has undergone and suffered and has come out alive. Now, they are able to draw the blood sample and collect the plasma, because these plasmas are rich in antibodies that has been able to overcome the viral antigen. Now, these are purified and transferred to people either who are exposed to COVID and who need the treatment, or doctor, nurses, healthcare professional, medical practitioners who need to have a prophylactic effect of not falling sick due to COVID. So this is a very large scale version of taking that monoclonal antibody and purifying it and transferring to needy patients as well as healthcare providers. A lot of people ask, like, what about the cure for COVID and how they are? The coronavirus that we have is a very unique. It is based as a radiating disk which have a lot of spikes on them. And by analyzing the morphology of this virus, the spikes can be bound to a cell wall and they rapidly multiply because the viral DNA is actually a RNA. So as per the central dogma, the RNA replicates itself, overcomes the body's mechanism, defense mechanism, and overwhelms it. So how do we prevent COVID? Yes, there is these antibodies, which are Y-shaped proteins that binds to the spikes that are present on the coronavirus. And by binding to them, they are able to prevent and inactivate it by adhering to the cell wall. This is very particular and useful in the current situation that the world is currently in. So the first step of any biological development, as I mentioned, is the antigen antibody and producing the monoclonal antibody in billions and billions of cells. So what we use is the CHO cell or Chinese hamster ovarian cells. These cells are very adaptable for growing in a suspension and ideal for large-scale production. And the second thing is that it can be serum-free and chemically animal-free media supplements can ensure that it is reproducible as well as repeatable results are produced from batch to batch. And most importantly, it allows post-translation modification of these recombinant proteins that are compatible with human beings. And there are several selective amplification that can be done using the CHO cells for higher yield of the recombinant protein per cell. So a single CHO cell can produce millions and billions of recombinant protein, the antibody that is needed for treatment of the disease. And what you see here is from my graduate school days, where I did extensive research on fermentation, large-scale production, and purification of protein. This was done almost 20 years ago, but it is still fresh, as you can see, because the technology and the building blocks of any fermentation process remain the same. These cells are grown in bioreactors. A bioreactor is basically a stirred tank mixer that has an agitator, certain sensors such as oxygen, pH, temperature. It has a mixer paddle and as well as a sparger that introduce oxygen for growth. And then you have a bottom discharge for removal of cell debris as well as effluents, the byproducts of cellular fermentation. So media, air, mixing, heat, pH, temperature, and nutrients are some of the critical process parameters that are involved in making any successful fermentation batch. And what are some of the design requirements that goes into a fermentation batch process manufacturing? The number one and critical aspect is the productivity and yield. In many cases, we are working with a large-scale operation wherein we have variability in temperature, pH, nutrient concentration, as well as process reliability in terms of reproducibility, as well as the yield that we obtain. The product purification after the batch fermentation is very critical, and it is handled separately as downstream processing. The most important aspect is when you have a nutrient media, you have oxygen and you have the ambient conditions necessary for cell regeneration and growth. There is a propensity for contamination, either bacterial or chemical, that can disrupt the output and yield of the batch. So we need to be very careful, and a lot of the manufacturing, if not all, is done under sterile environment. And there are different classification such as ISO class 1000, 2000, and 8000. Now, as you have seen in the previous slide, there is an enormous amount of energy that is required to keep the system in motion. Those are mainly the heat transfer of a jacketed kettle, mass transfer of a paddles, and momentum transfer, heat, mass, and agitation associated with the oxygen diffusion and media transport from either the top to the bottom or from right to left within the bioreactor. There is a whole engineering subject on reactor design and chemical synthesis that the chemical engineers have studied. This goes back to my original slide where you have the three main components of molecular biology, pharmacology, as well as the chemical technology that predates it. And last but not least is when you are producing these fermentation, you create bio waste. Those have to be treated, and state and local regulations need to be followed in waste treatment and disposal. Now, coming to our seminar on the QbD of biologics, there are many different functions that are associated in the fermentation process. And the most critical are highlighted here below. Some of them are temperature, the time, and the media. And the responses that you get are the yield, milligram per liter, the viscosity, and the molecular weight. So some of the key critical aspects when comparing a new to an old process is related to the oxygen uptake. So when you have like a comparison and you do a DOE trials, as mentioned in our previous presentation, DOE is just a part of the quality by design. DOE is not equal to quality by design, as mentioned by Paritosh. It's one of the tools. And there are other tools by which you are able to analyze, such as ANOVA and statistical process control, to determine whether the new system that is being implemented helps in the better transmission of oxygen to the biomass. And by running a series of trials and measuring the oxygen, you're able to show that there is a statistical difference between the old process versus the new process, which involved a box plot that is shown and represented here below. Although the averages are nearly identical, closer to 2, but you can see there is a significant shift in the percentile and the quantile values for the new process. And then another tool that can be used is the contour plot that is available. So you take the contour plot of yield versus temperature and time, because when you have three main factors, temperature, time, and media, we are isolating time, and temperature to see the maximum yield that can be obtained. Yield anything greater than 80% in a biological process is very fantastic. And by seeing this map, you are able to graphically infer that you need to be at the center, and a yield of 80 and over is obtained when the batch process runs for 88 hours at a temperature between 75 to 77.5. Now, when you look at the second response of the molecular weight and you plot a contour plot, our ideal monoclonal antibody molecular weight is in the range of 3,300 to 3,400. And you have like an operating band wherein you neither want something that is too high, neither you want something that is too low. You want to harvest the cells at the optimal molecular output of the monoclonal antibodies. So a contour plot is able to provide you that range where we previously saw 75 to 77.5 was optimal in the previous slide for the yield. However, in order to obtain the necessary molecular weight of the monoclonal antibodies for harvesting, you need to be between 77.5 to 80. Now you come to a concept called design space. When you have an overlay of the yield, viscosity, as well as the molecular weight, it gives you a narrow window of operation. The lines that you see are the limits with the operating curves. And the white spot, what we call it as the sweet spot, where you can maximize the yield and the output, is shown below. And you are able to widen this range or narrow it based on the limits that you set up. So in the blue intersection with the green line, you're able to see a narrow window between 77.5 to 80, whereas when the temperature is in the range of 70 to 72.5, you have a wider operating range. So this is what is called as a design space in the DOE trials that is obtained by using an overlaid contour plot of the yield, the responses 1, 2, and 3. Now, what is the third input variable that can be used? We had discussed about the media, the nutrients that goes into the media. So in this particular case, you can see that we are doing a series of experiments to see the effect of nutrient 1 and nutrient 2 at two different concentration levels, 1 and a 5. So this is a classical 2 by 2 factorial design that you run in replication several times, and you randomize this to see what the results that you obtained. And then you're able to get a net output with the concentration, saying that with 1 gram of the nutrient being added, your output is 0.93 and 1.27. Whereas at 5 grams concentration, with both the nutrients, you see 1.37, as well as for the nutrient 2, you nearly see a tripling at 3.1. And when you plot the graphs and see what are the values, you can see that there is a marked difference in the cell growth and output with nutrient 2 at 5 gram concentration as compared to the 1 gram as well as 5 gram concentration for the nutrient 1. So this shows that by using statistical analysis, graphs, and plots, you're able to make an inference about what would be an optimal range for obtaining the maximum yield and output. And this leads us to the last part of this QbD presentation, wherein we have a discussion on the interaction effects. So we talked about the three variables that are being present: time, temperature, and the nutrient concentration. And how do they act, either synergistically or interactively mild, or is there a strong interaction? When you have a linear system and you model that, when the lines are parallel to one another, you know that there is no interaction. Whereas a slight change in the media concentration can have a tremendous effect in the output of yield alone, as one of the responses is called a mild interaction. This is what we saw in the previous slide. But in cases of a strong interaction, where it's a simple 2 by 2 factorial process, you have the graphs crossing one another, results in very strong output, and you're able to get a narrow band called a sweet spot, where you're able to maximize the process as well as get the design space established. And these are some of the 3D predictive modeling that can be done using different interactions that can be seen between temperature, the nutrient concentration, and time that are used. You can use orthogonal design that are interactive and that use the diagonals, or you can use a simple face-centered design that just looks at the periphery and then you factor in to your design space. So to summarize today's presentation on quality by design in biologics development, we discussed about and learned about the recombinant DNA technology and how we are able to identify target sites of the virus or the tumor cells, analyze the morphology, and create the cell line that needs to be developed for producing that monoclonal antibodies. There are the upstream process that involves the reactor design, nutrient concentration, time and temperature for maximizing yield. And then there are the downstream processes that involves purification, as well as the qualitative testing methods that are there to analyze the monoclonal antibodies that are being produced. And invariably, you need to have a suitable media, which is in the form of an injection, intramuscular injection, for the drug product to be delivered to the human beings or animals. So a very critical aspect is, as I mentioned, concept to commercialization. I had shared with you some of the small batch size kilo scale reactors that are produced. But when you go to large scale manufacturing, the scale varies to maybe 1,000 to 2,000 liter batch sizes. These are commercial scale reactors. So scale up and tech transfer is very critical, that acts as a bridge between the R&D, where the innovation is being developed for new treatments, as well as the commercial manufacturing that can produce repeatable and reproducible results. So a lot of expertise, especially from engineers, pharmacists, pharmacologists, and microbiologists, is required for this, bridging the gap that exists in the molecular development to the commercial synthesis of this monoclonal antibodies. And another trust area that is upcoming in innovative technology and design are continuous stir tank reactors, CSTRs. This is part of the trust area related to continuous manufacturing by rather than having just one bioreactor and focusing on it, if you have a series of bioreactors and are able to do the processing and filtration in one step, then it minimizes the cycle time that is required from seeding of the inoculum to harvesting of the cells to production of the drug product. And this is one of the main areas that the industry is currently focusing on is the continuous manufacturing by having a series of stir tank reactors and producing the amplification that is needed of the monoclonal antibodies and filtering and obtaining the end drug product for human use. So rather than to go up with large scale up, such as in the previous slide that we saw with 1,000 and 2,000 liters, you can also minimize your footprint and have modular manufacturing by having a series of continuous stir tank reactors that are in optimal size of 100 to 200 liter scale. And with this, I would like to conclude my presentation. And if there are any feedbacks, questions, or comments, you could send it as an email to me.
Thank you, sir. Thank you for your excellent presentation. Now, along with your QA session, we'll also make it as an open forum or a panel discussion with along with the other two resource person as well as our senior professor, Professor Nagaraj Garu. I think we can take up that questions from the WhatsApp, which we have shared with you, and we can answer it one by one.
Can you please read the first question so that I can give you my answer as well?
Yes, yes. Yep. So the first question is regarding how to apply QbD in drug metabolism, or factors to be considered during drug metabolism.
Yeah, so the aspect of that is outside the scope of this current work because of this seminar, because our focus is on producing the biologics that is required for the analysis. Typically, the metabolism of the drug is done by the pharmacologist in analyzing that, and they come up with what are the metabolites that are needed. But in the biologics form, it typically refers to the antibodies that are being present. So when we look at the landscape, and I mentioned this in my slide, you can see that for the coronavirus, you have identified the molecular geometry of the spikes that are present on the virus and what are the suitable antibodies that are able to bind and prevent this virus from replicating. So that being said, in the biologics process, the metabolism or metabolites are out of scope because you're more focused on the suitable antigen and the antibody that can treat that, bind it, and prevent it from multiplication.
Now, the second question is open to all the resource person. So any one of you can kindly answer this. How to get good 5% LSD values on DOE? Are there any special considerations?
So I would like to answer this, and it's a very good question, and it's just an argumentation of what we have, what we learned yesterday, that this LSD values are like an indication of how variability is there in our project, in our considerations, predictions, or while doing the experiments, and then anything which we can do to minimize those variations, right? that would be helpful in bringing down those LSD values. So it depends upon the design you choose, the factors you choose, the ranges applied, and the unexplained variations like lurking variables which we are talking of. So effective use of blocking, doing a randomization, a proper randomization, having a sufficient number of replications, especially for the center values, will definitely add and give us proper LSD values. Even if that we did have a large LSD values, we need to go back to our design and see if something more. I mean, do we need to change the design, or we really need to add more number of experiments to do that?
Thank you, sir. And now the third question is how to interpret the response surface methodology graph and counterplot in DOE. And the participant has also requested you to demonstrate using a free trial software, something like Design Expert or a MATLAB or any other suitable trial version. So they have been asking you for a demonstration. I think Mr. Kargowskar kindly consider the request.
I'd surely be. I mean, but at this point of time, we don't have the time and
Yes, sir.
Sometime
later.
Take it up in another class, and it really needs some time. At least an hour or 2 hour session to actually go through that. Now, as far as you said about how to read the response surface designs. So one thing I found, like see, the details are in the ANOVA itself. If you are data prone, but the 3D plots and response surface and the contour plots, they should ideally be used for the visual inspection. They are good. They are visually looks good, and we can just identify, say for example, if there is a curvature or not in the design, or if there are interactions or not visually. But all these can be perfectly seen when you go through this ANOVA table. All this data is available there. So for prediction, always we should use those ANOVAs tables specifically, and just to present as a visual thing, we should be looking at the 3D plots and contour plots. So my suggestion would always be, if you want to predict something, go very deep into the data, the ANOVA tables for each and for whatever designs you are using it. And for visualization, these 3D plots and contour plots are very nice to present. And especially if you want to see the interactions or not, right? They are visible there, but they are also there in the ANOVA table. So the p-values of AB square or ABC, the factors will tell you whether the interactions are significant, not significant. So my take is go with the ANOVA table to actually study the data, and for visualization, go for the 3D contour plots.
Thank you, sir. The next question to the panel of experts is, what is residual lack in the fit? Residual lack of fit in the RSM? Does it create serious impact with our model or design?
So, if I would like to take it up, see, see, what does the name suggest? Lack of fit. And as a layman, if you just hear this statistical term, it means that your model you selected is not able to handle all the data points. So lack of fit, and if it is a significant, simply means that just go back, relook in the data, look at the design. This design should not be used for prediction. So yes, if your lack of fit is significant, just go back and look into the design, how we can improve that. There are a number of ways that can be done. And one thing which we discussed yesterday, like while the features of designs, like introduction of or incorporation of center runs and the replications, these are very important. Lack of it has to do with the estimation of the pure errors, basically. So if you don't have the replications for the center runs, if you don't have the enough center runs in place, then your pure estimates would be either 0 or very less, and that actually hamper your lack of fit as statistical estimates. So if that is significant, relook at it needs to be worked on, and that should not be used for any predictions. So am I able to answer this, or like if you want more explanation? Yes. Actually, there's a lot of calculations involved. So if the participants or anybody like to understand, there are a lot of data available and a lot of calculations available online. If anybody needs to understand in more detail, they can write it to us, or like they can just type in and just go through online with the available resources, SAS, Minitab, and design experts. And there are calculations involved, and that would be helpful in understanding this calculations behind those lack of it.
Thank you, sir. The next question is,
as a thumb rule, if lack of it is significant, just drop this idea.
Yes. So the next question is to Dr. Bhaskar Rao, sir. Sir?
Yep.
So what kind of skill set or hands-on experience is needed at this particular hour for the new going to be graduates to attain? Or how do we need to fill the gap between industry and academics? And how can we become industry ready after college? What is your suggestions?
It is a very good question actually to answer, so it's overall, so the first point I would like to advise and suggest to the people in the academia and in the industry that effective academic industry interaction is highly recommended. So something like, I know Bapatla pharmacy, so you select a particular region and pull out the educational institutions, and you can call for any seminars from the industry. So on a particular topic to understand particularly about the workflow, many people are struggling to understand about how the industry looks like, what is the real workflow from the beginning to the end. So in order to understand from the workflow from the beginning to the end, so I think you can conduct frequent seminars by effective industry and academic interaction, maybe I think quarterly or half yearly or annually. So I would like to suggest, so like this only in USA, we always have a good interaction between the academic and industry. That is the first one. And the second aspect is, I would like to advise the students to acquaint with, equip with a good amount of skills and tools, which is very, very important, before going to the industry. So in my previous experience, I have seen that many people who doesn't know how to make a data presentation in the Excel sheet. So try to understand, try to well equip with typing, advanced Excel sheet to plug in the data, and SAS tools, JMP jump softwares. So, and also, these are very critical to understand before going for the, so likewise in the lab also try to understand and handle the HPLC equipment, machine operations. These are highly required before going for the any company, because if you go with any blank, it's very difficult actually, so nowadays. So keeping that industry academic interaction, and also try to keep abreast of the latest current trends, what is going on in the industry. For example, in the small molecules, in the larger molecules, what are the real current burning topics going on in the industry. For example, in the small molecules, we have seen a lot of blend uniformity is the critical one, which is going on in the burning as a burning topic. Second one is, in the case of novel drug delivery systems like liposomes, stability is the main important parameter. So try to understand this workflow and current burning topics by inviting the industry speakers. So, quarterly or half yearly or annually. I know it's very difficult to, invite for each and every college in India. So I would like to select a particular region like, either district, and try to organize the seminars, symposiums / workshops. That is the one. And the second is, try to interact also with our excipients companies like, vendors, which are very, very critical. So for any industry, there are raw material suppliers like, Calorcon, JRS, like equipment suppliers like, I know from here in USA, we interact with Natuli, Feti. So these are equipment vendors, excipient companies. Try to interact closely with them and invite them, and that way what exactly going on on the burning topics. This is very, very critical. So ultimately also try to see in the simulation models available in the YouTube, like the database SAS, JMP software, and also like how to forecast the marketing for any product portfolio by using IMS database, and also new port for the new drug or DMF filings. The next point is try to also go to the online for the ICH website and FDA website. You see tons of lot of data is available to understand about the workflow, particularly the CMC sections, biopharmaceutical sections, and quality assurance, regulatory. So by seeing all these things, definitely you will be in a position. If you start from the third year, I think third year first semester or third year second semester of your undergraduation, if you start by the time you come to final year, you will be in a position to well equip yourself to face the challenges. Above all, the current scenario in the industry, academia, so the people are facing a lot of issues and a lot of stress is going on. I would like to advise to the current students, try to develop your personality and effective communications, presentation skills, okay, by following the correct personality development. This is only possible. I suggest you to go to like, yoga, meditation, breathing exercises, like to organize, quarterly or half yearly, like, conduct these type of sessions in the, in the colleges. I think with this, I hope I answered your questions overall.
Thank you, sir. Then the next question is to Dr. Vengat. Sir, how significant is going to be the innovation on small molecules? What is its future?
Yes, small molecules will continue to be there. When you talk about innovation, what has been the innovation in terms of the drug product? We have tablets that were manufactured millions and years ago that are being still consumed in a different dosage form. People used to eat or treat themselves with bark of trees and roots and shoots and flowers as a treatment because they are particularly rich in a particular alkaloid or a chemical that gives you the therapeutic effect. In terms of the new dosage forms that are coming up, we have tablets, we have thin film, which are edible films, we have orally disintegrating tablets, as well as buccal or sublingual. So these are some of the current hot areas of innovation that is taking place in terms of the drug product. In terms of the drug substance synthesis, we have like new innovator combination products, as well as excipient and API mixtures that are being prepared. For example, we have premixed paracetamol with excipients that are from drum to hopper to tablet form. So when we talk about innovation, it is very critical for us to understand what it is. US is the leading example of an innovation-based economy, because once you are in the innovation mode, whether it was the jet engines, the internet and the computer revolution, or the wireless and mobile technology that is available, you jump from one curve to the next curve, and that is a very quantitative leap. In pharmaceutical, because of the complexities involved with the regulatory requirements, the innovation is more on the drug dosage form and the presentation. The new innovation that has been coming up is related to the monoclonal antibodies, the biologics, for treatment of certain specific diseases such as cancer, oncology, or HIV, that can be targeted and precise. And that is where the innovation is based on, is to how to come up with new innovative drugs to treat diseases that are currently there or upcoming diseases or pandemics such as COVID that came out of nowhere. And we need to come up with the product innovation to have the treatment established.
Thank you, sir.
Does that answer the question?
Yes, sir. Thank you very much. Now I request Professor Nagaraj Garu to add to this particular session with his suggestions or his advices to the young faculty members as well as students who are listening to this QbD session. Professor Nagaraj Garu.
At the outset, I am happy to be part of this webinar, and I've been listening to all the speakers for the last 3 days, and I should appreciate the organizers to bring in such a high-profile speakers of varying fields and to be in contact with our young budding pharmacists. And today, I was very much carried away the Dr. Venkataraman's Venkatrakrishnan's speech, and you had an extraordinary voice, sir, and and even I not got up to have a water also because I don't want to miss a small point. And your set induction to the topic is very good. You have gone back to the basics so that the students amongst can catch up, and you have brought them rightly into the track. That was really fantastic. And also to bring to your notice, we don't have a pharmaceutical biotechnology as a many colleges except Andhra University and Manipal and few colleges like that. In India, we don't offer M.Pharm biotechnology as a specialization. Very few students are taking that. There is no much demand for that. That's why many colleges have not started. But I'm sure the students who are listening to your talk definitely now will be motivated to take up that particular specialization. And of course, there is a lot of scope in large molecule research, where how you are the what I should say trendsetter, and you're the founder, and you are doing extraordinary very well on your own front. And also, this should be a very eye opener, where the interdisciplinary nature of the pharmacy, where we can take up any challenging tasks, where at this time of situation, as you rightly mentioned, and also you brought in the corona also in between, so that made very much interesting about how the large molecule research is going to be very, very important at the point of time. And this type of like yesterday I was listening to Partosh Singh, and where he has rightly pointed out that carry the technology, and catching up with the technology is very important. And is with the Bhaskar sir is also mentioning that, which is just now previously, the students and young staff members who are listening to the webinar, please take the cue from these three eminent speakers, who with a great difficulty we have got them, and they are miles away and spending their valuable time to share their experiences with us, and that is commending. And your message and your success, your expertise definitely will go a long way in making our Indian pharmacy students job ready, or industry job ready, or I should say market ready for taking up the challenges. I take this opportunity to thank mostly the organizers behind the screen, excellently coordinating the whole work, and also the YouTube. Yesterday I saw the YouTube again, how the talk has come. It was very good. And unfortunately, the YouTube, we don't have a mechanism to interact. So the speakers accept in the form of the messages, but otherwise it is one way, but it has come very well. And probably we may have to recommend the students to listen to you before coming to our classes with respect to DOE, with respect to large molecule QbD, and so on. So this really a good session. And my final word is thank you very much for all the three of you, and also the organizers of this involving me. And it's a really great experience. And and listening to all successful people like you, and you are doing excellent in your job, and sitting away miles, and your concern to the pharma youngsters in India, and therefore, it is going to be very, very nice to listen to you and take up the things where that should go a long way for the youngsters to catch up. Thank you for your
Thank
time.
you very much, sir. Thank you for your. Thank you very much, sir. Now I request our principal to address participants.
Persons, and very good evening to Indian participants. Am I audible?
Yes, sir. Yes, sir.
So I wish to thank all the three resource persons. Generally, as a teacher, in order to explain the mechanism of oxidation, we use it to say initiation, propagation, and termination. And here I can describe it as initiation, propagation, and promotion. So this topic is nicely initiated by Dr. Ryan Bhaskara, who taught the basics of quality target profile and what is critical quality attributes, critical material attributes, and what are critical process parameters, and how to correlate them. And that is the beginning of this QbD. Next session, the day 2, Partoshin Ji explained what is the need of multivariable analysis, why we should not do one factor at a time analysis. Because the advantage of DOE and optimization was nicely presented by Partoshin Ji. We are very much thankful to Partoshin Ji. And the promotion of this QTPP DOE aspects was explained by Dr. Venkat today by using mechanistic model. Because until unless mechanism is not known, how a biological product is developed, we cannot identify the key parameters. That may be a physicochemical parameter, or a pharmaceutical factor, or a process parameter. So these fundamentals which are required for promotion of yield of biological product was nicely explained by Dr. Venkat Garu, and we are very much thankful to all the three industrial experts. And at the same time, I cannot ignore the value of the senior professor from Padmavati Mahila University, who is well-wisher and good friend to me, Dr. Nagaraj sir. What is the importance of these concepts in academic institutions and how to perform these QbD aspects in various fields of pharmaceutical sciences, like product development in pharmaceutics, and how this QbD can be exploited in case of method development of HPLC, and similarly in case of a herbal product, the selection of the extraction conditions and its optimization, and in case of chemistry, the synthesis of drugs. What are the factors involved in synthesis of drugs, identification and optimization, and all those things was nicely touched by the senior Professor Nagaraj sir. I am very much thankful to you. And I have to say one more thing. The day 1, after completion of this program, I received a phone call from one of my beloved teacher. That's our is Dr. V. Murali Balaram sir, and presently he is working in Sultan Ulum College of Pharmacy as professor. He appreciated. At the same time, during discussion, I learned 2 topics during that note. One is the virtual reality, because we are now shifted to traditional class to virtual classroom. And we have heard about virtual labs in engineering as well as pharmaceutical technology. But what is the reality of these virtual classes? The faculty are taking classes, e-classes or online classes or virtual classes. How we can monitor the students, whether they are actually involved in the classes or not. So virtual reality will play an important role. And other one is the augmented reality. And those 2 are coming in the pharmaceutical industry also. So within a short period of time, we may go for such kind of technology. Because in case of QTPP or QBD, we have to identify some factors, but later those factors efficiency, that is the relationship between independent and dependent parameters, should be confirmed only by in vitro experimentation. And that experimentation, in vitro experimentation, precision and accuracy can be promoted by simulation. And what is the reality of that simulation? And that can be better to have such kind of knowledge, promotion of such kind of knowledge, and sharing such kind of knowledge to the students will be useful. And based on that, I realized where we are, where our students are there. The technology which you are teaching, which you are learning, and the technology which is used in industry, there is a gap. And QTPP, maybe within a short period of time, it may be replaced by another sophisticated technology. Our students will get a knowledge on QTPP. By that time, industry is going to adopt some other technology. So in order to provide such kind of knowledge or platform, what I have to do? Immediately, I called with that approach, Dr. Bhaskar. So I want to incorporate such kind of things to our students, not only our college students, entire pharma students, as well as pharma fraternity. Because though we are faculty, though I am having 25 years experience, but even then, in some of the fields, I am a student. Because there is emerging and advanced technologies, advanced concepts. So everybody has to update their knowledge. Everybody is a lifelong learner. So one of the start to the pharmacist will be provided because of lifelong learner. And every teacher is practically lifelong learner. So in order to address such kind of issues, what help, whether is there any industrial expert to teach such kind of aspects, then again, with the help of Paritosh Singh, we got one expert who is working in Mumbai in the pharmaceutical industry, who is having vast experience in computer simulation, pharmacokinetic modeling, as well as computer simulation with respect to pharmaceutical product development. And we hope within a short period of time, we are going to conduct that kind of webinar, that again, the international webinar with such kind of components. And I shared this idea with Dr. Nagaraj sir also. Sir also accepted to participate, to coordinate that kind of webinar. I am very much thankful to Bhaskar Rao garu, Paritosh Singh ji, and Dr. Venkat, and Dr. Nagaraj sir, and Dr. Murali Balram sir for sharing such kind of ideas to me to promote such kind of webinars in order to reach the students, because any program outreach is important. So in order to promote this program, the ultimate objective is how we can benefit our younger students, how we can update ourselves. So in order to address such kind of issues, this is the platform. And the last, I will to express this program, and I wish to express my sincere thanks to the organizing committee. And whatever the success of this program, I wish to dedicate my organizing team, which includes the coordinator, Dr. Sai Kishore, convener, Dr. Satish, and the computer assistants and technical assistants is given by John Sagar. And we are very much grateful to all of you. And I cannot ignore the success of this program is because of patient listening of the participants. So thank you one and all by giving this opportunity. So wish you a good evening to all of you. Thank you.
Thank you, sir.
Thank you very much.
Thank you for your encouraging words. Sir, along with, we also want to add our sincere thanks to Indian Pharmaceutical Association Education
Sorry,
Division,
I ignored.
Chairperson,
Dr. Vidyadhar.
Dr. S. Vidyadhar, sir.
Sorry, I ignored.
And we are also thankful to our Bapatla Educational Society for providing us this continuous support to organize such quality events. And we also thank all the participants who are with us for past 3 days. We will be coming up with many more such events which are useful to them. We also thank the participating institutions, and our special thanks to all the 3 resource persons who has given lights not only on their expertise on QbD, but also they have given some emphasis on innovation, entrepreneurship, and also with some of our resource persons, we also got an idea of startups. Thank you. Thank you very much. Thank you, one and all. Have a good day, sir. Thank you.